Hi, so good day. So today we will talk about central dogma of biology. So the central dogma of biology topics will be subdivided into three parts. So we have two recordings and one synchronous class. So for the first part, and I decided to record a lecture video. So let's proceed. So what stuff is responsible for transmission of traits? Is it protein, chromosome, or DNA? So today we all know that DNA is the genetic material that carries information from generation to generation. But have you ever wondered how and when DNA was discovered? So what experiments and observations led to the discovery of DNA? So we will take a deep dive into the annals of history and try to appreciate the humble beginnings of molecular genetics. Yeah, about that, we are only going to handpick certain scientific milestones. Extensive explanation about all of what happened would be difficult. It would be quite a mouthful and, of course, time consuming. Okay, let us have a review. Let us have a recap. So, of course, we will mention Gregor Mendel, the father of classical genetics. So after his discovery, we have Thomas Hunt Morgan. So we already discussed and talk about Thomas Hunt Morgan, specifically his experiment about Drosophila, melanogaster, or the fruit fly. And today we will have a new scientist to discuss, which is Frederick Griffith. But before that, let us have a quick recap of Thomas Hunt Morgan's contribution. So, aside from the experiment about Drosophila, we have to look back as well on the theory proposed by Theodore Boveri and Walter Sutton. So, we have the chromosome theory of heredity. So, as I've said a while ago, it was proposed by Boveri and Sutton. So, ano yung kinalaman ni Thomas Hunt Morgan dito? So, Thomas Hunt Morgan worked on inheritance and genetic linkage in the fruit fly, Drosophila melanogaster, provided incontrovertible evidence for the proposal. The unifying theory stated that inheritance patterns may be generally explained by assuming that genes are located in specific sites on chromosomes. Okay, so at least that is the gist of Thomas Hunt Morgan's contribution to the chromosome theory of heredity. But still, it was uh, a debatable topic during that time because most of the scientific authorities still believe that proteins are the genetic materials of inheritance. Well, in fact, during that time, they should have listened to Walter Sutton and Theodore Boveri, along with Thomas Hunt's substantial evidence. Now, we have Frederick Griffith. So after Thomas Morgan, we have Frederick Griffith. So Frederick Griffith was a British bacteriologist whose focus was the epidemiology and pathology of bacterial pneumonia. So in January 1928, he reported what is now known as Griffith's experiment, so the first widely accepted demonstrations of bacterial transformation, whereby a bacterium distinctly changes its form and function. He showed that Streptococcus pneumoniae, implicated in many cases of lobar pneumonia, could transform from one strain into a different strain. So the observation was attributed to an unidentified transforming principle or transforming factor. This was later identified as the DNA. So he was America's leading pneumococcal researcher. So along with Oswald T. Avery. So these two scientists speculated that Griffith had failed to apply adequate controls. Yun yung claim naman ni Avery. So thus, Avery and his team of scientists tried to improve Frederick Griffith's experiment. But before that, let's talk about the mechanics of Griffith's experiment. Okay, so just to give you an overview, this is the series of steps that Griffith followed. Okay, pneumococcus has two general forms. We have the rough and the smooth one. Now first, let's talk about the smooth strain. So the S form is more virulent and bears a capsule which is a slippery polysaccharide coat or simply a mucus. So, ayan. Yung mucus na yun, pinaprevent niya yung mga immune cells nung host na patayin yung bacteria. Okay? Thus, nai-infect nung S strain yung katawan nung host and eventually namamatay yung host because of pneumonia. Now, dito sa first step na ginawa ni Griffith, makikita natin na meron tayong live S strain. So, in-inject yun dito sa isang mouse and yung mouse, after several days, namatay. Now, the second strain is the rough strain. The rough strain don't have a coat or a capsule. Okay? So, walang coat or capsule yung R strain. Thus, madaling patayin ng immune cells yung R strain. So, nape-prevent ng immunity natin yung infection. So, syempre, since malakas yung immunity, yung mouse, healthy siya. So, hindi siya namatay. Now, we have the third step or the third setup, which is an S strain is heat kill. So, syempre, patay na yung bacteria, ibig sabihin hindi na siya ganun ka hazardous or ka-delikado na i-administer sa isang mouse. So, ang nangyari, in-administer yung heat killed and nabuhay yung mouse as expected. But the fourth setup is a surprise because we have live R cells, and using the live R cells, heat-killed S cells or concoction of heat-killed S cells is added. Now, surprisingly, the mouse contracts a pneumonia and eventually succumb to death. So, ano yung nangyari? Bakit kaya ganun yung epekto? So, one of Griffith's conclusions is that there is something in the concoction of heat-killed S strain pneumococcus that transformed the rough strain. So, ayan. So the ability to infect the mouse with pneumonia was transmitted from S strain to R strain. So what is the agent of transmission? How did it happen? So ano kaya yung naging transmission? So ano kaya yung agent rather? So syempre, ito yung common na sinasabi ng mga scientists noon. It's a genetic material. But scientists during that time are still in a heated discussion whether it is protein or it is DNA. Okay? Or rather the chromosomes. So, protein versus chromosomes. Now, to make a improvement on Griffith's experiment, we have McCarthy, Avery, and MacLeod. So, criticisms about Griffith's experiment is that in that experiment, Griffith failed to show the molecular mechanism on how the transforming agent affect the rough strain. Okay? Okay, so let us explain the Avery, MacLeod, and McCarthy experiment. So we have a heat-killed S cells concoction here. So we have to remove the lipids and sugars from a solution of heat-killed S cells. So a method was used to extract these other components. And we prepared several combinations of concoctions. First, we have uh, a no protein solution. Third, we have a no RNA solution. And the third one is the no DNA solution. So for the no protein solution, we added enzymes. So particularly proteinase. So it's a type of enzyme that breaks down protein. Okay? So let me use my pen. So the proteinase destroyed the protein. So the first concoction is a protein-free solution. Then our cells are added to that concoction. After several days, the R cells or the rough strained pneumococci was transformed into S cells or the smooth strain. Now we have the second concoction. Another enzyme was added, which is RNase. So RNase dissolves the RNA. So in here we have a concoction made up of protein and DNA. So yun po. So yung RNase, winasak niya yung RNA. And itong concoction na to, linagyan siya ng rough cells or rough strained pneumococci. And after several days, still the same, meron tayong S strain na nag-appear. Ibig sabihin, yung R strain na linagay nag-transform into an S strain. Still, the genetic material is present. So remember the end goal or the purpose of this experiment is to isolate which type of material affects the transformation of these cells. So for the third setup, we add DNase or an enzyme that destroys DNA. So the third concoction was no DNA concoction. And we added an R cell. Surprisingly, this time, there is no transformation. The R cells are retained rather. So ibig sabihin, na-isolate na natin yung genetic material this time. So it was DNA. Kasi, since wala na yung DNA dito sa concoction, ibig sabihin, siya yung transforming factors. So remember, yung DNA present dito sa previous setup. Merong DNA dito, meron ding DNA dito sa flask na to. Dito sa concoction na to. So DNA is the transforming factor. Okay po, so hopefully nakukuha natin yung concept. So pinasimple na natin yung explanation. So after the experiment of Griffith, Avery, McCarthy, and MacLeod, we have the following scientists: Martha Chase and Alfred Hershey, then Rosalind Franklin, as well as Francis Crick and James Watson. So let's proceed. So we have the Hershey-Chase experiments. In 1951 and 1952, Alfred Hershey and Martha Chase conducted a series of experiments at the Carnegie Institute of Washington in Cold Spring Harbor, New York, that verified genes were made of deoxyribonucleic acid or DNA. Hershey and Chase performed their experiments, and later named the Hershey-Chase experiments on viruses that infect bacteria, also called bacteriophages. So the experiments followed decades of scientists' skepticism about whether genetic material was composed of protein or DNA. And the most well-known Hershey-Chase experiment, called the Warning Blender experiment, rather the Waring blender experiment, provided concrete evidence that genes were made of DNA. So the Hershey-Chase experiment settled the long-standing debate about the composition of genes, thereby allowing scientists to investigate the molecular mechanisms by which genes function in organisms. Now, to perform their experiments, Hershey and Chase utilized a technique called radioactive isotope labeling. So, chemical elements can exist in different structural forms called isotopes. So, isotopes of the same elements are nearly identical, but scientists can distinguish between them by experimental means. One way to differentiate between chemical elements with different isotopes is by analyzing the radiation. So some isotopes are less stable than others and give off radioactive signals. Uh, and these radioactive signals are detected. Okay. Now, some isotopes are less stable than others and give off radioactive signals. So Hershey and Chase marked phages by incorporating radioactive isotopes of phosphorus and sulfur in those phages. So we have two particular elements. We have sulfur and we have phosphorus. So remember these two key elements. So they allowed some of phages to replicate by infecting E. coli that scientists had grown in radioactive sulfur. So the researchers rather, let other pages infect and replicate in E. coli that scientists had grown in radioactive phosphorus. So, ganito kasi yun. So, di ba sa DNA, ang meron lang tayo is yung phosphorus. So, walang sulfur sa DNA. On the other hand, sa protein, meron tayong sulfur. Merong presence of sulfur. So, gumamit tayo ng radioactive isotopes nung dalawang magkaibang element para ma-detect natin. Okay, kung ano ba talaga yung genetic material, whether it is the protein or the DNA. Okay po. So, gamitin na natin tong image na to sa pag-explain natin. So, dito sa batch 1, let me use my pen. So, dito sa batch 1, meron tayong flask na may bacterium. Meron tayong culture ng E. coli. And itong E. coli na to, in-infect natin siya ng bacteriophage. Yung bacteriophage, yung meron siyang radiation ay yung protein coats lang. Yung DNA niya walang radioactive isotope, walang label, walang isotope label sa DNA. Instead, yung may isotope label lang po ay yung protein. Kaya makikita nyo dito sa label, radioactive protein. Okay. Now, the concoction will undergo a centrifugal process. So, yung paglagay natin ng concoction sa isang centrifuge will result to a separation of materials based on their densities. Of course, materials with less density will be at the top, and materials with heavy or more density will be at the bottom. They will become the precipitates na tinatawag natin. Okay? Now, dito, pagkatapos ng centrifuge, makikita natin na meron tayong precipitates. Itong mga pellet na to. But upon looking at the pellet, hindi radioactive yung mga pellets or yung mga E. coli na nandito. So, the precipitate is made up of E. coli bacteria. On the other hand, ito naman, dito sa taas, ito yung protein nung protein coat rather nung mga bacterias. So, suspended yung mga protein coats. So, ayan. Kung naman sana nung E. coli bacteria yung radioactivity, di sana ito yung radioactive na part. Itong mga precipitates, itong mga pellets sa baba. Next, we have batch 2. So, itong batch 2 naman, meron tayong concoction. But this time, ang radioactive naman is yung DNA. So meron tayong isotope label para sa DNA, which is the phosphorus, the isotope for phosphorus. So it will undergo the centrifugal process. And makikita natin dito na yung liquid part, the less dense part, of course, nandun yung mga proteins. But this time, hindi siya radioactive ones. Kasi walang label yung proteins na to. Walang isotope label. So ito naman, dito sa pellet na to, meron dyan mga E. coli na may mga radioactive DNA from the virus, from the bacteriophage. And this bacteria replicates. So kapag nag-replicate yung bacteria, yung sumunod na bacteria, yung mga bagong bacteria, taglay niya yung DNA nung galing sa E. coli. So, nakuha na. At least yung gist nung experiment nila Hershey and Chase na ipaliwanag na natin. Now, this experiment, this discovery cements the position that the DNA is the genetic material rather than the proteins. Kasi yung proteins hindi naman itatransmit nung E. coli kapag nag-replicate siya. So, yung protein coat nung E. coli bacteria hindi naman radioactive after that. So, okay. Tapos na tayo kay Hershey and Chase. So, ang usapan ay yung history ng DNA. So, saan ba talaga nagsimula ang DNA? So, nung high school pa ulit-ulit na sinasabi sa inyo na si Watson and Crick ang nakadiscover ng DNA. But no. Ang discovery nila, Watson and Crick, is only the molecular structure of DNA. Okay, yung structure lang ng DNA, tandaan natin. But it was Frederick Miescher, or Miescher. I don't know how to properly pronounce this name. So, pasensya na if nabutcher ko yung name ni Frederick. So, in the 19th century, science was beginning to look at the origins of life through a microscope. So, Charles Darwin and Alfred R. Wallace gave us the theory of natural selection and evolution in 1859. In 1866, Ernst Haeckel, a German zoologist who sketched and named the art and nature, proposed that a nucleus might contain heritable components. And a decade later, we have Walter Fleming, a German biologist, and the fifth child following four sisters described chromosomes during mitosis. In spite of all this revelatory work on nuclear stuff, most championed the idea that proteins held the key to life. Little did anyone know that in a small laboratory in Germany, a quiet perfectionist with an eye for detail was discovering DNA, and that is Frederick Miescher. So, he was born into a family immersed in science on August 13, 1844 in Switzerland. Both of his father and uncle Johann F. Miescher and Wilhelm His, respectively, were renowned physicians, anatomists, and professors at the University of Basel. So, Miescher showed keen interest in science and took up studying medicine in Basel, but later decided that research would satisfy his curiosity about the natural world. So, ayan. Meron siyang sets of experiments. So, hindi ko na i-explain yung mechanism ng experiments. But basically, dun sa set of experiments niya, meron siyang nakitang precipitate dun sa solutions niya. So, the story of DNA begins in 1868 when Frederick Miescher joined Felix Hofsaler's lab. So, Miescher was interested in the chemical composition of cells. So, Hofsaler's lab was where biology and chemistry shook hands. And this environment provided Miescher with the right tools to go about investigating. Using pus, o kaya nana, diba? Pus or nana from fresh surgical bandages sourced from a nearby clinic. So, Miescher obtained leukocytes. So, his cell of choice, yung immune cell, the leukocytes. So, Anong ginawa niya dun? So the structure of DNA was elucidated in 1953, but it was actually discovered in 1868 in a small laboratory in Germany by the Swiss scientist, Mischer. Okay. When asked by most people with the simple question, who discovered DNA, we usually get the answer, I don't know, or Watson and Crick. So in most school level science textbooks, the names that pop up during the introduction to DNA are James Watson, Francis Crick, and Maurice Wilkins, the Nobel Prize awardee in 1965. So yun po. So in the 19th century, si Frederick Mischer ang nakadiscover. Wait lang. I'm thinking about the mechanism and how he discovered the the precipitates and the leukocytes. Okay. So ito na po. So since Mischer is meticulous in terms of recording every detail and working with great caution, so this patient approach allowed Mischer to detect something odd amidst the proteins and lipids of the leukocytes. So while performing tests, he noticed that substance precipitated in acidic conditions. So excited and intrigued, he set out to explore these unusual precipitates. Okay, he did several experiments to the precipitate and the substance out of the solution. So this unknown precipitate dissolved under alkaline conditions but precipitated when the solution was neutralized. Even when the solution was made acidic, the substance did not dissolve back into solution. So, ayan. So, di ba? Ganito kasi yan. So, kapag acidic yung solution, usually, natutunaw agad yung proteins, di ba? Na kumbaga nagbe-break yung mga bonds ng peptides na nawawasak yung protein. But the structure of DNA is different. So during that time, nung ineksperimentohan ni Frederick Mischer yung leukocytes, so nagkaroon siya ng mga precipitates dun sa kanyang specimen. So sinubject niya yun sa iba-ibang klase ng acid and other substances. And hindi agad-agad naapektuhan ng acids. So instead, na-retain yung integrity. So, fast forward, itong substance na to, pinangalanan na DNA or deoxyribonucleic acid. Kasi iba yung nature niya sa protein, kaya ganun po. So, si Frederick Mischer ang unang nakadiscover. Hindi sila Watson and Crick. Molecular structure lang yung na-discover ni Watson and Crick. Now we have Rosalind Franklin. Now the quest now is to find the molecular structure of the DNA. And during that time, Rosalind Franklin was one of the leading expert in radioactive crystallography. Okay, so he is a British scientist, best known for her contributions to the discovery of the molecular structure of deoxyribonucleic acid. So a constituent of chromosomes that serves to encode genetic information. Franklin also contributed new insight on the structure of viruses, helping to lay the foundation for the field of structural virology. So, ito na yung storya niya. So, in 1951, Franklin joined the Biophysical Laboratory at King's College London as a research fellow. There, she applied X-ray diffraction methods. So, para mapag-aralan yung DNA. So, when she began her research at King's College, very little was known about the chemical makeup of structure of DNA. However, she soon discovered the density of DNA and more importantly established that the molecule existed in a helical conformation. So her work to make clearer X-ray patterns of DNA molecules laid the foundation for James Watson and Francis Crick to suggest in 1953 that the structure of DNA is a double helix polymer, a spiral consisting of two DNA strands around each other. So, he is famous for this photo. Ito po. So, this is photo number 51. So, the photo number 51 shows you the cross-section of the DNA. So, kapag ang perspective, yung pagtingin natin sa DNA is nandito, front view. So, suppose ito yung DNA structure, di ba? Mahaba siya. Horizontal. Now, kung titingnan natin yung DNA ng pag ganito yung perspective, nasa harap natin mismo yung end, ang makikita natin ay ganitong klase ng structure. So, itong X formation na to, yan yung pag-coil ng DNA strands. So, di ba merong dalawang DNA strand? Okay, merong dalawang strand ang DNA rather. So, itong dalawang strand na to, nagko-coil with each other. So, ito yung pattern na nakukuha natin dun sa coiling ng DNA. Okay. Now, sadly, Rosalind Franklin did not receive any Nobel Prize because he died, or rather, she died because of cancer. Ayan. And sino yung nakakuha ng credit dun sa discovery na to? Francis and Crick. Crick and Watson, rather. So, pasensya na. Medyo nabubulol. So, Nobels are only awarded to living scholars, but a handful of researchers and advocates in the past few decades have argued that Franklin deserves more credit than she got for the DNA discovery. So, her images of the DNA structure reveal DNA as made of two helices, rather. Okay? So, it was Maurice Wilkins, one of her fellow, that shared the picture or the photo 51 to Francis Crick and James Watson. So, hindi ethical yung pagkaka-obtain ng picture. Kumbaga, somehow Maurice Wilkins is stabbing Rosalind Franklin at the back. So, sinishare yung pictures nung X-ray crystallography dito sa dalawa na to. Kasi during that time, merong tatlong teams na nagko-compete for the Nobel Prize. We have Francis Crick and James Watson, and we have Maurice and Rosalind Franklin, and as well as another team from Caltech. Ayan. So, Maurice Wilkins shared the file to Francis Crick and James Watson. So, itong file na to very important kasi because of that file, because of that picture, Francis Crick and James Watson was able to find out kung ano ba talaga yung structure ng DNA. And eventually, although there are lapses in their discovery, because of the discovery of another scientist, which is Shergaff, na nakagawa na sila ng isang perfect model ng isang DNA. So, makikita nyo dito, wasak na wasak pa rin naman yung model na ginagawa nila. So, hindi pa masyadong buo. So, nahihirapan sila, Francis Crick and James Watson, na gumawa ng molecular model. So, it was because of the help of Rosalind Franklin and Shergaff that they were able to construct a good model. So, and that ends our lecture video for today. So, informations about Shergaff and his two laws or rules will be discussed on the next session, which is synchronous one, by the way. So, sige. So that ends the discussion. So thank you for listening. And hopefully, somehow, at least, naging clear sa inyo or nabigyan kayo ng background dun sa history ng molecular biology. Hopefully, ma-appreciate nyo yung history na yun. So that's it. Thank you for listening and goodbye.